Okay, so the first question is, which among the following is or are for mesenchymal stem cells? So there are some source of mesenchymal stem cells. So there are some tissues given here: adipose tissue, amniotic fluid, bone marrow, umbilical cord tissue. So the question is, which are all the sources of mesenchymal stem cells? So can any one of you answer? Okay, so I'll just say mesenchymal stem cells are usually otherwise called stromal cells that has the ability to self-renew, and also they exhibit multiple lineage differentiation. So mesenchymal stem cells can differentiate into a bone cell, a cartilage cell, a fat cell, neuron, anything. So they have a multiple lineage differentiation capacity. Just a minute. Yeah, so the some sources of mesenchymal stem cells are actually all these options are sources of mesenchymal stem cells. So other than these, placenta, bone marrow, breast milk, adipose tissue, the dermis, Wharton's jelly, dental pulp, and endometrium. All these are sources of mesenchymal stem cells. Which of the following is an example of multipotent stem cells? So the options given here are zygote, heart muscle cells, hematopoietic stem cells, and inner cell mass of blastocyst. So to answer this question, you should have known what multipotent stem cells are. So there are different potencies of cells. So can anyone say what they are? Any one potent cell type is also fine. Hello, am I audible? Hello?
Yes, ma'am.
Yeah, okay. Okay, fine. We'll look into the answer. So an example of a multipotent stem cell is hematopoietic stem cell. So let's see what cell potency is. So the different types of potency are totipotent, pluripotent, multipotent and unipotent cell. So totipotent cell is a cell that will give rise to a whole organism. Maybe if it is a plant, then one single cell that gives rise to a whole plant, or if it is an oocyte or a sperm which fuses together to form an embryo, that single cell is a totipotent cell which will give rise to the whole organism. Next, coming on to pluripotent cell, blastocyst, the inner cell mass and everything of the blastocyst is a pluripotent cell. So it won't give rise to a whole organism, but it will give rise to other cell types of the lineages ectoderm, mesoderm and endoderm. So cells which come under these lineages are multipotent cells. Example, the the cells that give rise to the neurons, that is all these stem cells. They are multipotent cells, and unipotent cells are fully differentiated cells. Example, the neuron, epithelial cells, cardiomyocyte, liver cells, pancreas cells, the specific cells, fully differentiated specific cells. Moving on to the next question. What are the functions of skin? Mention the functions of skin that are listed below in the given options. So the options are calcium production, protection against pathogens, provide fluid homeostasis, thermoregulation, vitamin D synthesis. Yeah, that's correct. Except for calcium production, all the other functions that have been listed here are the functions of skin. So protection against pathogens, we know that skin is a primary barrier, external barrier and the largest organ of the body, so it will provide protection against pathogens. Fluid homeostasis, it is involved in excretion of the fluids through the skin. Thermoregulation, which is regulating your body temperature, and vitamin D synthesis. So the last four options are some functions of the skin tissue. Okay, this fourth question is a question regarding the mechanical property of a biomaterial. So, which of the following statements is or are correct about stress strain and the relationship between them? So, I hope you would have heard about the stress strain curve, the stress strain relationship, that is a Hooke's law. So based on that, this question can be answered. So they are asking which of the following statements is correct about the stress strain relationship. So option A is strain is a force acting per unit area. Option B, stress is the fractional change of length of the specimen. Stress is the force acting per unit area. D, strain is the fractional change in length of the specimen. E, stress and strain are tensorial quantities. F, plot of stress versus strain showing an initial linear region in which the Young's modulus E is constant and a nonlinear region where increasing E, leading ultimately to fracture of the specimen. I will show the figure in the next slide. Option G, Young's modulus is defined as the ratio of stress to strain to stress, or is it defined as the ratio of stress to strain? So this is the diagram, and with this you can answer what are all the options given above are correct with respect to stress strain relationship. So let's see one by one. Strain is the force acting per unit area, or stress is the force acting per unit area. So usually stress is the term that is defined as a force acting per unit area, so option C would be correct. And the other way, for strain, it is the fractional change in length of the specimen. So option C and D are correct options. Stress and strain are tensorial quantities. So if you want to measure the mechanical property of a biomaterial, that is the tensile strength or a compressive strength, for all these properties you will need a stress versus strain graph. So stress and strain will give you information about the tensorial quantities. Next option, plot of stress versus strain, will first give us, in the next slide. Here you can see this region, you can see that it is linear and it is steadily increasing, and the slope, when you take the slope of this region which is linear, you will get the Young's modulus, or otherwise it can be called as the elastic modulus. So the elasticity of a biomaterial, if you want to calculate that, you will get when you take the slope of this region. So when you stretch a particular material, for example if you take a rubber band, when you stretch a rubber band, it will, to one extent, it will have elastic region. The elastic region will be to an extent where it can regain to its original shape. After that extent, it will undergo a deformation which is not the elastic, which will not be in the elastic region but in the plastic region. So when a deformation goes to the plastic region, it can't be brought back to its original shape. So this is the elastic region, and this part is the plastic region, where the particular material can't be brought back to the original shape, and this is the fracture point where the material finally breaks. So this is the stress strain graph. So it will always be y versus x, that is stress versus strain. The slope will give you the elasticity of the particular material, and this highest stress, this particular point is the ultimate strength, that is the ultimate stress it can withstand in this particular strain percentage. So when you increase the strain percentage beyond this level, the amount of stress it can withstand will start to decrease, and ultimately it will tend to fracture. So C, D, E, F, and the last option that is correct is Young's modulus is defined as a ratio of stress to strain. So y by x. So I have given a plot here which will give you a rough idea about why this stress to strain plot is being used for biomaterials. So if it is a plastic material, the elastic region will be very less and the plastic region will be more, because when you stretch a plastic material, the tendency of it to regain its original shape is very less, so the elastic region is very less, and if you take a slope, the value also will be very less, the Young's modulus value will be less. If it is a ductile material, it will be elastic to one particular region. After that, it will start to have a permanent deformation in its structure. If it is a brittle material, it can withstand a very less strain percent and it will immediately break. So the fracture point of a brittle material is very less. So for example, if you take a rubber band, it will have a high elastic region. If you take a glass, it will be a brittle material and it will easily break at a very less strain percentage. So this is the an overview of why you should study stress to strain for any biomaterial. So for example, if you are going to prepare a material for bone tissue engineering, your bone should not be as elastic as a cardiac tissue or a muscle tissue will be. So a bone will have a certain tensile strength, certain strain that it should withstand, certain stress that it should withstand. So according to those properties only, you should fabricate a material for bone tissue engineering. So based on different tissue types, all these properties will differ, and only when you are able to mimic the actual tissue types and its properties, you will be able to then only it would be meaningful to engineer a tissue. So yeah, this is it. If you're having any doubts, you can ask. Okay. Okay, I'll move on to the next question then. What are the applications of tissue engineering? So this is a multiple select question. Anything that is not an application of tissue engineering, you can exclude. So can anyone answer this question? Repair of damaged bone tissue, development of 3D bioprinted organs, engineering lab-grown meats, and removal of diseased tissue. So which are all are applications of tissue engineering? Yeah, that's right. So A, B, and C, these first three options are applications of tissue engineering, and removal of diseased tissue is nothing but just medicals. So can I have you all heard of lab grown meats? Okay, so tissue engineering, apart from engineering tissues that are for human purposes, engineering a tissue that is used for edible purpose, that is the concept of lab grown meat. So what they do is exactly what we do in a human tissue engineering concept. So they will take, for example, if you want a chicken, they will isolate stem cells from the embryo of a chicken, and they will grow those stem cells in the lab and differentiate them into muscle cells, that is myoblasts. So they will differentiate them into muscle cells, fat cells, that is adipose cells, and different cell types, which they will co-culture again and then form a fully grown flesh out of it. So this is one concept that is being experimented. I mean, it is a current advancement in tissue engineering that has been used to that will be used as an alternative for the meat that we are eating. So it has various advantages. Even though if it's costly, it is one advantage that has already been. There are many startups working on this, and there are also products that are being in sale of lab-grown meats. So this is one application, a current advancement in the field of tissue engineering with respect to other animal tissue. So other than that, repair of damaged bone tissue is for tissue engineering, and development of 3D bioprinted organs are a typical example of tissue engineering. Moving on to the next question, what are the bone forming cells called? So fibroblasts, osteoblasts, neuroblasts, and myoblasts. So among these, which are the... Yeah, it is osteoblast. So other cell lines, myoblast is a muscle cell. Neuroblast is as you all know, it is a neuronal cell. And fibroblasts are normal skin cells. Keratinocytes, fibroblasts are all skin cells. They are usually present in all the lining of a tissue, of an organ, fibroblasts. So the answer is osteoblast. Does anyone of you know what an osteoclast cell is? There are two types of cells, osteoblast and osteoclast. Okay, so osteoblast cells are bone forming cells. Osteoclast cells are bone resorbing cells. So the function of osteoclast cell is to break down bone tissue, while the osteoblasts will form new bone tissue. Why do we need osteoclast cells? Like, we don't want to break down do we want to break down the existing bone tissue? I mean, what will the function of an osteoclast cell be? Okay, I'll explain it in an easy way. So, for a tissue, to engineer a tissue, you need cells and a substrate for the cells to grow. So, cells in our body will be attached to the extracellular matrix. So, the function of an extracellular matrix is will be like a substrate on which the cells will multiply, proliferate, differentiate, and so on. So, whenever a tissue is being formed, you might have heard of the term tissue remodeling. So the ECM that is present in the tissue will help in formation of new cells also, and it will remodel itself so that the old cells are being the apoptosis of old cells will happen and formation of other new cells will happen, because the tissue can't be static and the ECM can't be static, because you're growing day by day. You need space for the new cells, the new proliferated cells, to be occupied in the tissue. So tissue remodeling concept is the concept that will come under that. When you want to remodel a tissue, you need cells that will disrupt and digest the existing cells also. So in bone, there are two types of cells like this: osteoclast and osteoblast. So osteoclast cells will resorb the bone cells, and osteoblast will help in formation of new bone cells. Moving on to the next question. The movement of nutrients and minerals from higher to lower concentration gradient is known as dash. Diffusion, osmosis, peristaltic movement, or abduction. Okay, so it's diffusion. This is a very general terminology, but then why we use this term in tissue engineering is that, so we prepare a lot of materials. These materials may be polymers, ceramics, metals, and carbons, and so on. So whenever you prepare a material, you will be doing it with a lot of chemicals. So for example, if you want to prepare a biomaterial with PLA, polylactic acid, or if you want to prepare it with polyethylene glycol, that is PEG. All these are synthetic polymers. When you are taking a synthetic polymer, you have to cross-link these polymers first to form a mesh, and that cross-linked polymer should be converted into a scaffold or a hydrogel to mimic the tissue ECM. So when you're doing all these processes, you will involve so many chemicals with that. So to balance the pH, you will be using HCl and NaOH, and to increase its mechanical property, you will be using many chemical cross-linkers and so on. So you can't directly prepare a material with so many cross-linkers and so many chemicals and directly implant it into the body. So there will be a lot of immune rejection that will happen in the body. Even if you place it on a skin surface, you will definitely see inflammation for this material. So in order to purify the material, if you're adding HCl or NaOH, definitely there will be salts and water that are being formed. So once you have prepared your material, you have to put it in a dialysis bag and you have to purify it. So during this purification, you will need this process of osmosis or diffusion and so on, where the diffusion is a movement of minerals or nutrients from higher to a lower concentration gradient. Once you have purified your material, you can easily use that gel. You can inject it inside in situ gel, or you can use it as a tissue for an excision wound. So the answer is A. Moving on to the next question, which of the following is the most abundant protein in the human body? Yeah, it's collagen. So collagen is the most abundant protein. And do you know what gelatin is? You guys would have heard of gelatin also. Is gelatin found in human body? Yeah, correct. So gelatin is not found in human body. It is just it is like we artificially prepare it. So it is just like a substitute for collagen. You can't isolating collagen from a tissue is difficult, and you want since collagen is present in the human body in a higher percentage, you want a material that will mimic the properties of a collagen. So that is how gelatin was found, and gelatin is being used almost invariably in all the biomaterials that are being synthesized nowadays. If you take any article regarding biomaterial or tissue engineering, basically for the cells to attach, you need proteins, and you need moieties, the RGB domains you need in your material for cells to attach and grow and proliferate or differentiate and form a full tissue. So for that you need RGD domains, and these RGD domains are found in abundant in this gelatin. So gelatin will definitely be used in almost all of the hydrogels or scaffold constructs that you will be studying in this course. Okay, so the next question, the advantages of autographs include. So what are the advantages of autographs, or what are autographs? Yeah, yes, that's absolutely correct. So autographs are uh, tissues that we are taking from from individuals of ourself actually. So it will be our own body cell. So are graphs taken from twins also called as autographs? So if there are identical twins, and if you want a graph taken from another twin, is it called an autograph, or? Yeah, okay. Yeah, that's correct. So, the advantages of autographs are all the options: less or no graft rejection at all, easy availability of donor tissue, less or no risk of disease transfer. So, one example of autograft that I can give you apart from the examples that you would have seen in the video is that, so usually when a skin tissue is engineered for plastic surgery, plastic surgeons usually prefer skin that is being taken from any other part of our body and replaced it in our face. So other than that, autografts are also grafts that are made from, if you take a if a person is having a burn injury, you can collect his blood, and you can the blood will have all these components: plasma, which will have plasma, and the RBCs. So when you centrifuge the blood, you will have the RBCs collected in the bottom, the fibrin which is collected in the middle, and the top part is the serum. So a plasma without platelets, clotting factors are serum. So you will get these three layers, and you can prepare a platelet rich fibrin material from the middle layer of a centrifuged blood sample, and that material also can be grafted on the patient with a burn injury so that the skin tissue can grow on top of it. That is also one type of a graft that is being prepared from the patient themselves. So since it is prepared from the blood taken from the patient, there won't be any rejection. Ultimately, definitely there won't be any rejection. So it is being nowadays it is being practiced as one type of a biomaterial, not a biomaterial that is prepared from the patient's own blood. Yeah, so that is all for the introduction class. So these are some of the questions that will help you answer your week 1 assignment, you would have all seen the videos. I hope you would have all seen sir's videos. And if you guys are having any doubts regarding the videos or the live session that I took now, you can ask. So if I just want to know if this is very basic for you, or I can even go deep into the concepts of tissue engineering. I want to know, yeah, I think almost all of you are from biotech background here. So I want to know if you are able to understand the concepts, or if I should explain more deep. Okay, fine. Okay. So if you're having any questions related to this live session or the videos, you can put it in the forum. Myself or the other TAs will reply to your questions. I hope it's being answered now. Any other doubts? Any questions regarding the course contents? Or if you want the assignment questions to be solved once the week is over, also please let me know. I will give you clarifications for all those questions. Yeah, so is this the question that you asked?
Ma'am, you told something about autografts and that person with burnt case after that centrifugation. So after centrifugation, what is it?
Oh, okay, that one. Yeah, so the thing is, when you collect your blood and when you centrifuge it, it will form 3 layers. The bottom layer is the RBC, and the middle layer is the fibrin layer, and the upper layer is the serum. So what the practitioners, that is the medical practitioners, what they do is they collect the fibrin layer that is being formed, they pour it in a petri dish, and they add some cross-linkers that will clot all the fibrin tissue that is present in the blood. So it will become into a material. Within 2 to 3 hours, it will form by itself, it will form a material, and that material will be taken and it will be restitched onto the burnt tissues of a victim. So if there is any burn victims, the burn wound, over the burn wound, it will be restitched. So one of the project that we are doing here is that in the next class, I can show you some pictures of it for better understanding. So that what I wanted to tell was that is also a graft that is being prepared from the patient themselves. So it is also like an autograft. So since it's for wound healing purpose, we will also load antibiotics onto the material like gentamicin or tetracycline, and then we will stitch the material directly, suture it on the patient. Yeah, so the difference between plasma and serum is, yeah, so a blood will have a solid component and a liquid component. So when you centrifuge a blood, all the solid component will settle down. Other than that is the liquid component, and in the liquid component there will be these clotting factors, the thrombocytes, the platelet cells. So when you exclude the platelet cells from that liquid portion, that part is called a serum. A plasma is nothing but serum plus the clotting factors. So a plasma is on the whole the liquid portion, and when you exclude the clotting factors alone, then that is the serum. Am I clear? Yeah, so a Matrigel, okay, Matrigel is a patented product that has been developed from. So I think you guys would have read about Matrigel. It is actually developed from. It is an extract of all proteins and small molecules that are present in mouse tumor, and they have taken that and they have derived, decellularized it completely, and they have prepared a material out of it. So since that material will contain all the collagen, laminin, all the extracellular matrix components that are required for a to engineer a tissue, all that will already be present in that material, matrigel. So it is very easy to take a matrigel and then culture cells on it, instead of us preparing an altogether a new scaffold or a hydrogel and culture cells on it. So matrigel is a tested and patented material that is available. And that is why it is being very commonly used in tissue engineering. So all the characteristics, I mean the physical characterization, chemical characterization, biological characterization, the cell viability potential of the material, everything has already been characterized for matrigel. So it is easy for us to prepare a scaffold or a hydrogel directly from matrigel and grow cells on it. But the concept of tissue engineering is I mean, the scope is more than that. So matrigel is, as it is biocompatible and it is very easy to culture cells, but then the cost of it is also very high. So always there is a scope for finding out better materials which are cost effective and but still give the same properties as a matrigel. So that is why we are working on or experimenting various other natural proteins and polysaccharides so that we can use them to prepare an alternative. Yeah, so from the next session onwards, maybe I will discuss questions, the assignment questions, the previous year questions, and maybe the last 10 or 15 minutes, I will just give you an overview, just one slide about one article, like every class, so that you will know what is the exact application of the contents that you will be studying in the course. Okay, so if there are no more questions, then we can end the session here. If you want to discuss anything, you can stay back. Thank you. Thank you.